talk about Post-oposSOM tools, next level transcriptome analytics. Blockbuster title. When I prepared this talk, I actually thought this workshop will be like 20 people and all of them more or less familiar to what we do and what we have done to our projects. Then I heard, okay, there will be 150 or 70 or how many of you are now participants. Then after some hours of thinking, it also came into my mind that like 90% of you will have only one question, which is, what the hell is opossum? So I now want to show you some details. Hans teased some of our analysis results, and I want to give like a little overview from a more conceptual point of view. So, but the very first thing you have to know is about the problem itself. So what's our problem? We are more or less data scientists. So everything is data. It's more data and it is more data. So when you start to look at your molecular data in Excel sheets, also you are completely lost. You can apply very expensive cryptic programs, and you are only restricted to functionalities which are provided by these programs. So this is one problem, and you can subsume this problem more schematic. We have a large number of features. They can be like abundances of proteins or metabolites, expression values of genes, methylation values, whatever. These features are in a vast number available using modern experiment techniques. But now, especially with the rise of single-cell sequencing, we have a very large number of samples available. So formerly known as the large P small n problem, we now have large P, and oh my god, it's a real large number of samples. So this is one problem. So I have to have in mind a huge number of features and a huge number of samples for one data set. But I don't have only one project with one data set. I have a multitude of projects and data sets I have to have in mind. And not only me, but also Lydia has several projects and data sets. Maria has several projects and data sets. And our boss, Hans, he also wants to keep track with our things we do, so he has all of those data sets in mind. So this is a challenge, how to effectively perform analysis. And the solution for this is more or less a software development process. In the very beginning, we have to find an approach which is like suiting for us. And it was very easy. We just take the best sorting machine available on market, which is the self-organizing map. It's like the truck Hans showed us. It has a lot of advantages. It's a very simple sorting machine in principle, like this device my son used to play with 7 years ago. So now we have this approach, and in a 10-year lasting process, we developed a workflow. We try to make it very efficient for us and comfortable for us, that we can work very good in our institute. And the outcome of this software development process is the so-called opossum pipeline. Hans also showed some functionalities from. And in the opossum pipeline, there is a lot of functionalities, and we subsumed them to the term data portraying. So data portraying has some properties, very important properties. So it's based on a primary analysis using such a self-organizing map, a SOM. And this map is simply a grid, and the machine sorts features. These features could be, for example, expression profiles of genes. The machine sorts those profiles according to similarity. The next property, or the next feature of our data portraying, is that we have individual visualization of the samples and of the groups. we have a modularization of the data, we have function mining, and actually we have a lot more implemented in this pipeline, but I want to restrict today to these points. So the first point is the so-called expression portraying. So Hans already showed those mosaic pictures. So for one random sample, we get such a picture. And you have to have in mind that at each pixel is one position in this map. At each position in this map, there is a cluster of genes with a very similar expression profile. And this location is fixed. So you can see here in this map blue and red regions, which refer to underexpression blue and overexpression, which are the red pixels. And as I said, we have one self-organizing map. It will be trained in the beginning of the opossum pipeline. So that means we have one coordinate system. One pixel is always a fixed set of co-expressed genes, and the staining, the red and the blue color, is according to the expression of one sample or one group. And what we see here is basically the transcriptional fingerprint of this sample. So we generate for all of the samples in a data set such portraits. This is again the lymphoma data set, lymph node cancer with about 1,000 samples. But you cannot simply look at 1,000 such pictures. You will not find anything. Again, it's like staring into an Excel sheet. Same value. So the next step we do is a data modularization. That means each red region you can find here will be collected in some kind of master map. And here these red regions are so-called modules of co-expressed genes. And these modules can be attributed to some subtypes, because I can simply look, okay, this subtype is so-called Burkitt lymphoma. They all show, or many of them show, those 4 red spots here, and that means all of them can be attributed to the Burkitt lymphoma subtype, and so on. Between these spots, we can calculate co-expression, or like frequency or probability of mutual expression or simultaneous expression, and this results in this co-expression network Hans also showed before. But we need to know, what does it mean? Okay, they have those red spots, they have a module activated, but we need to know the biological function behind that. So we perform an enrichment analysis. We have a database of it's not some thousand, it's now already 10,000 of sets of genes with known biological function. And we can look into each of those modules. We know which genes belong to this module, and we can see which functions they belong to and which functions are enriched there. For example, I told you here, this is one module which is activated in the Burkitt lymphoma subtype, and we also know many of replication, DNA proliferation, and so on processes are activated, or are the genes enriched here, and they are activated in the corresponding subtype. But not only sets of genes can be analyzed on the functional view, also single genes. So that's also a map Hans teased in his talk. We know where each gene is located in our coordinate system, and then literature tells us some 100 genes which are reported to be mutated or broken or whatever in lymphoma cancer subtype, and we map some of them into this map. So one gene I learned to know in the last years, this is SMYRC, it's a proto-oncogene. It is located here in this module, which is belonging to the Burkitt lymphoma again, and it enhances proliferation. So that's why also proliferation is associated to this module. So data portraying resulted in actually 11 papers which have portraying or portraits in the title. We were able to publish it in quite good journals, and if you ever wanted to find the logo of this workshop. You can find it here in this paper from this year. But not only 4 papers we have. In total, we have 38 papers using opossum parts, opossum functionalities, or offsprings of opossum. Not only we apply opossum, also other scientists, so we have about 3 citations per year from people who actually mostly write an email, hey Henry, can you help me? I have a problem with opossum. And a year later, we get a citation. So also quite good journals. But the question is, okay, now we have opossum, but what comes after? So there is interested researcher A, like Siras, with some bioinformatics skills. So he downloads the opossum package from Bioconductor, applies it, analyzes data. He will be happy, and we will get a citation with maybe some intermediate communication. That's very good. Then we have interested researcher type B, asking questions like, can you reproduce your results with independent data? Could be like a reviewer of a paper. So we want to have the portrait, module annotation, function annotation, molecular subtyping, everything we did with the opossum pipeline, but now on new data. Okay, one solution could be to simply run opossum again and use some training. But the problem is, okay, we have now worked like 2 years with one data set. We have learned these spots belong to this subtype and so on. We have learned what the functions are which belong to each of the modules. But now we start a new sum training with the own data. Then we paste the independent data to this and get completely new portraits, landscapes, modules, everything is new. So we have a new sum training, we get new landscapes, new modules, new molecular classes. Another problem is that systematic bias between those 2 data sets are enhanced by the sum training. So actually, this is game over. This will not work. That's why we came up with a solution for this problem, which is called extension sum. And the extension sum trains new samples or independent data into an established sum landscape. So you can see it here. We have part of the data which is our own data, the previous data, and we have part of the data which is independent data, maybe downloaded from the internet, or maybe some new samples generated in additional experiments. Then we apply the so-called XSUM, extension sum training. The original part of the data will participate in an active type of training, and the new part of the data will be passively trained into this map. Then we can run all of our opossum pipeline functionalities, like portraying, modularization, function annotation, classification of the samples into molecular classes. But we also have, for our independent data, in the same coordinate system, portraits and new molecular classes. So that means if I see in one new sample that this spot module here is red, the module is active, I can simply look into my master map, can see, okay, this is also activated in diffuse large B-cell lymphoma, DLBCL. It also fits to the annotation I downloaded with the data. I can see, okay, these and that functions are activated, and it could belong to this or that molecular sample class. So, for interested researcher number B or type B, we can provide this extension sum. And it is very important, and I have to repeat that the new samples will be mapped into the established expression landscape. So, and interested researcher type C. He has also questions like, where is my beloved gene whatever? What's the expression profile? What about function whatever? What is the difference between subgroup A, subgroup B, and subgroup C? So you can see we have a basically unlimited number of variations of only a limited number of question schemes. So we can solve, or we solved it in the last years with a lot of programming efforts, but actually this is could be very nicely solved with an interactive tool. So instead of having the data, running opossum, and letting opossum generate 2 GB of files for you, we simply use the opossum image. It's just a workspace image, put that into a Shiny app, and what we get out is the so-called opossum browser, which I want to show you now, live action demo. So this is the opossum browser. We have implemented already some 3 data sets for you. And one part of this opossum browser is now the gene browser. So this is a list of all of the genes measured in this data set. So if any one of you is now interested in a particular gene, name it. 10.
P10.
Yeah. Now it's retention receptor. No, I basically only know like 10 of genes. Yeah, let's say Myk. Now, let me see why. Myk. Yay. Here we got the Myk gene. And as I developed this app on a screen which is like this ratio, we have to scroll now down, and we can see the expression profile of this Myk gene in the different classes, and we can see where it is located. This is now the gene browser, and very similar layout we can see in the gene set browser. Arsene, name any.
Carrot.
I heard immune response.
Okay.
What is this? Come on. So we see there are 336 genes in this gene set. They are distributed over the map, but they like enrich or accumulate in the top left region. We can see here in which samples or in which classes the genes which belong to this gene set are overexpressed. So this is very nice because any researcher could now go to this browser and look into the gene or in the gene set browser, looking for his favorite genes or gene set functions which he is researching on for years maybe. Now, very nice thing is the phenotype browser, which is also part here. On the right hand side we see this correlation network. Each of this bubble is one sample in the data set. That's again more or less the picture we see here. And now if I have a special question like, where are all the young guys? I go to age and I can see all of the children and adolescents are located here. And if I want to know what's different to people having this cancer in older age, I can simply select them. We see they accumulate more in the lower part, and we have also here the survival curves. We can immediately see if there is a real impact of this phenotype for prognosis. So with this tool we can also handle a lot of different questions, and the last tab I have here is the pathway flow, pathway signal flow, PSF, which is a method developed mainly here in Yerevan by Arsene and his group. It is a very nice method which allows a detailed view into the flow of the signal in the pathways. So, for example, we can now look into B-cell receptor pathway, signaling pathway, because we have B-cell lymphoma. It's very interesting. So, actually, a very nice control is to check what is the signal flow in the healthy B cells. And we can see orange or brownish colors means they are more or less all active. That's what you expect. Then we can see for each of the genes which are here, we can see where it is located into our SOM map, and we can also see what is the signal value in the different subgroups. So this is a very comprehensive tool, and it we get rid of a lot of individual programming, the same line for like 100 genes we were asked. So, where's my talk? Okay, so this now brings me to conclusion. So for type A researchers, we have the opossum pipeline available. For our language, it is publicly available in the bioconductor repository. You can download it and use it. For type B researchers and for reviews of papers, we have the new extension SOM, and we can perform all of our data portraying functionalities for new samples or independent data sets, and it will be performed in the same coordinate system, in the same expression landscape we are used to and which we have worked with for a longer time. And for type C researchers, we have the interactive clicking tool, and I think if real lymphome researchers would use it, they would really find hidden treasures in dataset we have not crossed yet. So if you want to try it out, here you can see the address, and you can go to this page. So I'm finished with my huge opossum advertisement. Thank you for your attention.